Good morning, everyone. Thank you for joining us. First, I would like to thank Alison Walton from the Immokalee IPCs Center for her help and cooperation. Today's program offers 2 CEUs for pesticide license renewal and 2 CCAs for crop advisors. If you need CEUs, email me your name, email address, and phone number. The program sponsors are Tom Johnson with TJ Biotech LLC and Meg Wally with 3 Defender LLC. They are paying for your refreshments and lunches if you are attending in person. As you all know, researchers at the University of Florida have been looking for solutions to HLB from many angles. Today's seminar program will focus on 2 topics. Number 1 is life after individual protective covers, maintaining health of citrus trees in the long term. The speaker is Dr. Fernando Alferez, Assistant Professor in Citrus Horticulture, UF-IFAS, Southwest Florida Research and Education Center in Immokalee. Topic number 2 is what you need to know when using trunk injection to manage HLB. The speaker is Dr. Albrecht, Associate Professor in Plant Physiology, UF-IFAS Southwest Florida Research and Education Center in Immokalee. Fernando, it's all yours.
Thank you, Mongi. Okay, good morning. I want to start this presentation today with an overview of what we will be talking about. I will talk first about what we know about IPCs now. Then I will discuss the potential of brasinosteroids to protect young citrus plants recently planted in the field. Then I will discuss what we are doing to see if we are able to combine both tools, IPCs and brasinosteroids, to protect further the trees. And then I will show you some data that we are collecting now about varietal differences when using IPCs, because what we are seeing is that not all varieties behave the same under IPCs. And we have some very interesting data on that. So this is a slide that I have been showing several times, but I'm putting it today here because you see that we have 2 different states here, young trees and mature trees. But as we advance in this work, we are getting a 3rd state of the matter in this case. So we know that we have mature trees that they are 100% infected by HLB, and they are declining in production. But at the same time, we are very active planting healthy from the nursery, healthy trees that are HLB free, and they are not producing yet. So the goals are totally different in both cases. In the case of mature trees, of course, we want to maintain the trees productive, and if possible, improve the health and improve fruit yield and quality. That's the major goal. In the case of young trees, to keep them free from disease until they enter into production age or younger. What we are having now is a 3rd stage, and we will talk about that later, having young trees that are reaching that production age healthy. And then how can we prolong that health in those trees? And that's most of the talk today is going to be about this. Well, the truth is that unfortunately, this is the rate of HLB infection in a non-protected, newly planted citrus grove here, at least in Southwest Florida. By 6 months, we have 80% of the trees testing positive for HLB. Okay, and in 1 year, 100% of the trees are already infected. Now we know that IPCs, individual protective covers, are a successful tool in protecting young trees from the psyllid infestation and then protecting the trees from HLB infection. And you know the mechanism is really simple, it's psyllid exclusion. We know that because all the grower experience, but also because of the work that we did here at SWFREC. And just as a reminder, we started this research 5 years ago. We planted several trees, actually 90 trees, of Valencia on Cleopatra rootstock, and we kept those trees for 30 months covered with IPCs. Those trees bloom for the first time inside the IPCs, but then after 30 months, we removed the covers. Okay, we harvested 6 months later in February 2021, and then we harvested again in February 2022. So from August 2020, the trees were exposed to infection. So we've got 2 harvests already on those trees that are now 100% infected. And by the way, next week we are harvesting our 3rd season from these trees. So we will be comparing the yield and quality in these trees that are already 100% infected from at least 1 year. Now, the first and the most important result that we got is that IPCs, as we have been showing several times, prevented Silence infection. Just as a reminder, this is real-time PCR values. So we are showing the CT value. Everything above 36 cycles means non-detectable, or there is no bacteria in those plants. Everything below 36 means that we are able to detect the bacteria. So you can see that trees covered with IPCs were testing negative for these 30 months, as compared to the trees that were not covered from IPCs, with IPCs that were testing more and more positive during this period of time. Not only that, the growth in these Valencia trees was greatly improved with this tool, with the IPCs. And this is once we remove the cover. So this was after 30 months of cover. And not only that, also the area of the leaves was larger in the trees from the IPCs. Okay, so this is what happened when we remove the covers. So in white, the white dots is the rate of infection that I told you at the beginning of this talk. By 6 months, you have 80% of the trees infected. By 12 months, you have 100% of the trees infected. This is when we install the IPCs, so that's the black dots, and you can see how you don't have any tree infected until we remove the covers by month 30. And then you get the same rate of infection, okay? And again, by 6 months, you have around, there was a little bit of delay, but still you have 60% of the trees infected at that time. Well, at this point, since I told you before, when we removed the covers, the trees were already blooming and setting fruit, we were more interested in looking into fruit quality. So, and as I told you, we harvested in February 2021 and February 2022, the second season. Remember, this was Valencia, and this was early February, so it was really early in the season. And this is what we found. We found that even though these trees were getting infected, fruit quality was improved by the IPCs. The first season was 6 months after IPC removal, so we got about 80% of the trees infected. And the second season was 18 months after IPC removal, so 100% of the trees were infected. And you can see how in both seasons we got an increase in bricks in the trees that were protected by IPCs, compared 10.9 versus 7.5 in the controls the first season. Then the second season, 9.6 versus 7 in the controls that were infected from the beginning. This picture on the left are fruit from the second season. This is the typical appearance of the fruit in this second season with the trees 100% infected. What we are seeing here is some decline in quality from the first to the second season, 10.9 versus 9.6. We don't know what's the trend in the third season. We will know this next week as we harvest. And I will be communicating that as soon as we know it. So that takes me to what I was saying at the beginning. Can we maintain this health and this fruit quality over time? So I think in the last 10, 15 years, this is a novelty in citrus in Florida at this moment. We are getting trees that are at production age that are free from disease for the first time in several years. And that's because we are using the IPCs. We remove the IPCs. These trees, at least in Valencia and Hamlin, because we have also some research done in Hamlin with some collaborators. But at least in these two sweet oranges, we are getting trees that are getting into bearing age and are free from disease. So can we keep those trees free from disease longer than that? And that's what we are trying to achieve now. What we are doing that, how we are doing that? Well, at the time that we were working with IPCs here at the center, we had also a trial working with brasinosteroids, which is a kind of novel plant growth regulator that has been used in several other crops with really good results, but never used before in citrus. And what we were seeing, so we were treating also young plants, mature plants as well, but I will focus now on young plants, and maybe during our discussion, if there are questions, I can address what happened with mature plants. But in young plants, we were seeing that there was a reduction in the rate of infection. So this 80% that you see here, that was the control trees, and that was a totally separate set of trees, totally separate from the trees that I was showing before that had this 80% infection rate. So the infection rate, if the trees were not protected, was very uniform across all our fields in the center. So we had an 80% of the trees infected, but when we treated with brasinosteroids, that rate was much lower. We were having a 25% of the trees infected by 6 months. So it was a really interesting reduction. So the question comes naturally: can we combine both systems? Can we work with IPCs until the tree starts bearing fruit, and then once we remove the covers, start treating with brassinosteroids, because one of the things that we did during all these years was to make sure what was the safe and the most effective concentration of product to apply to these young trees. So could we apply both tools in a combined system? So what we did was to apply to USDA NIFA for funding, and we got an award last year that will be allowing us to do this work, which is combining IPCs and brassinosteroids to try to prolong health and improve fruit yield and quality in newly planted trees. This project started last year. Here I'm showing the team of collaborators, all located here in this center. My colleagues, Dr. Sute Albrecht, Oscar Batuman, and Jawad Qureshi. And then we have also one postdoctoral associate working on this project, Dr. Sosan Ben Abdallah. And we are having a master student coming soon to work on this as well. So it's going to be a very nice work that we already started to do, because these are the objectives of the project, and I will show you how we are working already, how we started already working in the second objective. But the first one is just to study the effects of both tools and their combined use on the physiology of the tree and then fruit yield and quality, and also HLB incidence and disease progression. Now, the second objective that we already started working before, and I will talk about that a little bit, is to study the effects of brassinosteroids, specifically on biology and behavior of the vector, and on reducing incidence of pathogen transmission, and also finally to assess the effect of the combined system on other pests and diseases. And I will talk about that a little bit also, because that was something that was unintended when we started to do it, but we are seeing very interesting things. So as I told you, we started to work before on that, because I have a collaboration with a group in Valencia, Spain, from Institute of Valencia Agricultural Research, and they got a Fulbright grant to come last year to our center. So we started to work on this, because they are specialists on citrus plant immunity. Their lab works focus on this. So we started to look in depth into the immune response of the trees after brassinosteroid treatment. And this is the only pathway that I will show you. So this is the only one, but I want to show you this because this is a central pathway of immunity in plants and also in citrus. This is the corismate pathway. This pathway leads to salicylic acid, which is one of the central modulators of plant immunity. It has two branches. One is called the isocorismate branch. You have it here. And the other branch is called phenylaniline branch. Both compete for the substrate that is corismate. Now, when we treat the plants with brassinosteroids, it was very interesting to see that we got a massive induction of both pathways, the isocorismate pathway and the phenylaniline pathway. This is important because it shows that brassinosteroids is actually producing a strong downstream response, because both pathways compete, but both are massively induced. And as a consequence, we got all the downstream immune response heavily regulated by 8 times as compared to the control plants. So, and this happened just 5 days after the treatment. So the treatment was done in plants in cages, and what we did was to apply the treatment. Then we put 4 couples of psyllids, 4 males and 4 females, inside the cages. And we looked into their behavior and their the reproduction. And you can see that by 1 week already, this is ×0. This is 1 week later. Remember, after 5 days, we had this induction in gene expression, this induction in immunity. You can see how we were able to see less psyllids per flash. We are doing that as we speak. We are doing that also in the field because these were control conditions. But what was more interesting is that after 6 weeks, this dynamic continued, and we got still less, significantly less psyllids per flash. This was after 6 weeks, which is really important because I didn't say it at the beginning, but when we started this work with brasinosteroids, we were applying every other week the material because that was what a seminal study did with young plants. They were applying every 15 days. So we were thinking that probably applying once per month could be enough if we increase the rate, the concentration of the product. And we were seeing some results, applying just once per month. This, what is telling us is that maybe we can apply even less frequently than that, because after 6 months, we still have some psyllids, but this difference is significant. One result, one result that was totally unexpected was the effect on armyworms and moths. You know that inside the IPCs, armyworms are one of the main problems, and is really difficult to treat. So the problem is that the moth lays the eggs on the outside of the cover. Then the caterpillars, when they get born, can get inside the IPCs, and they can eat the whole tree. That's a big problem. So what happened here was that we had an infestation of moths inside one of our greenhouses. And what we found was that the cages of the containing the trees that were treated with brasinosteroids had less eggs and less moths than the controls. That was unexpected, and that suggests us that probably the brasinosteroid treatment is producing something in the plant, in the tree. Probably it's changing the volatile profile. We are looking now into that because there are some reports that brasinosteroids actually can change the profile in leaves. So they are producing, as a part of the immune response, they are producing volatiles that repel insects. And in this case, this is what we think we are seeing. We are looking now into that. So we think that brasinosteroid treatment after IPC removal is a feasible possibility because the things are still healthy and young at that time when we remove the covers. And that's when it's described that brasinosteroids are more effective in producing a strong immune response when the trees are younger, 2-3 years, no more than that. Now, the data that we are collecting suggests that brasinosteroid treatment may still be effective in protecting the trees for more than 1 month after we apply. And this is important because that probably will allow to reduce the number of applications, and that will have an effect on the cost. And now what we are doing now is to work on knowing if brasinosteroids will improve fruit quality further, as we saw after we removed the IPCs, and if we can avoid the decline in Brix over time. We have some indication on that in mature Valencia, that we have seen that brasinosteroids are able to maintain Brix over time. And for the first time, we have seen that in Hamlin this year. We are seeing that Brix are maintained over time. So every time that you get an off bloom, for instance, and the Brix go down, that's not the case in brasinosteroids. They get stable over time. Brix are not great at this time, especially in Hamlin. We are talking about 8 point something, at least in this area. Brasinosteroids are maintaining this level. Now, the second part of my talk is focused on the differences that we are finding between different varieties when we cover those trees with IPCs. This is a study that started in January 2020. We planted 700 trees of 3 different varieties that we arranged in a complete randomized block design. And the varieties were Sugar Bell, Early Pride, and Tango. And they were grafted in 2 different rootstocks, US 942 and Sour Orange. And we removed those APCs just last December. So we have been collecting data every year, and I will show you just some data, because we have a lot of data, but some data from the end of the first year and from when we removed the covers in December, the 3rd year. So this is what we got in several parameters after 1 year. So in purple, you have the IPC covered trees. In greenish, you have the non-covered trees. And you can see that in general, we didn't see any improvement in early pride in actually in any of the parameters that we were looking into. But for instance, for the first year in Sugar Bell and Tango, we got much more content in chlorophylls, and also in the color of the leaves, and also in the height of the trees. In this case, early pride, yes, was a little bit higher also, but especially in Sugar Bell and Tango. And it was interesting, and we can discuss about that, what happened with the volume of the canopies. So we didn't find any improvement in Sugar Bell. We found a nice improvement in Tango, but in the case of early pride was the opposite. Having the IPCs on early pride was deleterious for the tree. Actually, they were behaving better without IPCs. So after 3 years, we removed the covers, and we produced a scale that allowed us to rate these trees once we removed the covers. This was after 3 years. So a number 5 would be a good productive tree, commercial tree. A number 4 would be still really good trees, and that they will bear fruit. Number 3 would be a tree that still have potential, but we need to work on those trees to rejuvenate them. 1 and 2, don't bother, they are lost causes, they are trees that are dying. And we had some zeros, that mean dead tree, especially in early prides. Or no tree, we had a hurricane last September, so those trees were not there anymore. You can see how Tangos, they were really good trees under IPCs. Still, Sugar Bell were nice trees, were almost close to 4, but not early prides. Now, I'm showing you some pictures of how these trees look like. So in the case of early pride, what we have seen is what almost everybody that has early pride is seen, and we have also early prides in our CAPS facility, and we have a lot of dieback. Well, this was under the IPCs, we had a lot of dieback. In the case of sugar bell, well, here you can see a comparison, a tree that was covered and a tree that was non-covered from the beginning. In general, we found larger leaves. The main effect, and probably the most desirable effect, was that all the trees were uniform in size. So the trees that were not covered, they were, there was a lot of difference in the sizes, but the trees that were covered with the IPCs all were uniform. But by the third year, the growth was really constrained, especially in the trees that were grafted on US 942. And also something interesting is that we didn't get any fruit set under the IPCs in sugar bell. This is a solid block of sugar bells. There is nothing around. So you can see how the tree that was not covered had a lot of fruit, because I took this picture in a couple of weeks ago, and you can see already some fruit drop because the fruit is senescing. But you have a decent amount of fruit in the trees that were not covered. That was not the case in the IPC covered, and that tells us something that we observed before, and is that these sugar bell trees need some pollination, not cross pollination, but pollination from other sugar bell trees. So summarizing this, our recommendation here is that don't cover sugar bell trees for more than 2 years, because after that you don't get any additional benefit. By first year you have an increase in the canopy. The second year that is maintained, but after 3 years you don't have an additional benefit. Now in the case of tango, it's totally different. So you can see here a covered tree and an uncovered tree. And here you can see a comparison of one of the replicate set of trees, and you can see how all of them are uniform in size. They have more dense canopy as compared to the non-IPC trees. They have this very interesting morphology that I had a visitor from South Africa last week, Paul Cronje from University of Stellenbosch, and it was the first time that he saw these trees and the APCs, and he just said this look like lollipop trees. So this is like a lollipop morphology that we know that it will disappear over time as it starts to produce new shoots and expand the canopy. And of course, the trees are taller. So in this case, yes, in this case, is a clear advantage. So in conclusion for this talk, the first one is something that we already knew, but I want to remind again. IPCs were able to prevent Silas infection in all varieties that we studied, even in early pride. Brasinosteroids, we have seen that are inducing a strong immune response in young plants, and they also, very interestingly, reduced psyllid infestation for at least 6 weeks after treatment, and this has the potential of reducing the frequency of applications. We are currently assessing this possibility as part of the project that I just told you. Now, fruit quality appears to start declining after the second year of IPC removal. What we are doing now is to look into the possibility of brasinosteroids as a way to prolong fruit quality after the IPC removal, but also other treatments, for instance, OTC injection, can come later as the tree start to decline further. We know that IPCs confer an additional horticultural advantage to Hamlin and Valencia oranges, sweet processing oranges, that translates into better fruit quality at least for 2 years. And also we have seen that in tango mandarins in terms of horticultural advantage to those trees. But we haven't seen any additional effect on sugar bell beyond 2 years of covering. In the first 2 years, yes. And early pride, we didn't see any better performance under IPCs for those trees. And this is all what I wanted to share with you today. Just thank you to the Tree Defender for providing the covers that we have been using in our studies, to the USDA for the new grant that we just started, to CRDF for continuous funding of these studies, to my colleagues here at SWFREC, Drs. Albrecht, Batuman, Qureshi, and Zekri, and to my team, my team that is doing a great job on this and other projects, Susan, Maria, Osvaldo, Murillo, Tim, and Divya. And thank you for your attention. I will take any questions.
Thank you, Dr. Alferez. We will start by answering questions from the Immokalee IFAS Center. Anybody here has a question or a comment? Go ahead, man.
This is the data reporting on the process relative to
Not in citrus. In other plants, yes, it has been shown that it may increase immunity and impact some other diseases. There has been a lot of work done on model plants that they don't tell us much besides that it can work, like for instance Arabidopsis. That yes, it impacts not only the salicylic acid pathway, but also the jasmonic acid pathway and also ethylene. And all combined, it has an effect on different diseases. But in citrus, there was the seminal work was done by some Cuban researchers like 6 years ago, and it was the only study on citrus, and the rest of the things, to the best of my knowledge on citrus, is what we are doing. So we haven't looked into other diseases. What we have seen, for instance, in citrus on our hands is that not only we have some repellents on psyllids, also on the moths that produce this problem, also on rust mite. We have seen a reduction in rust mite in the fruit when we treat with racinosteroids. Is the only effects that I have seen.
Any other question?
Yes, sir.
Wait, Jim.
Yeah, there's studies that indicate that jasmonic acid is upregulated by racinosteroids and gibberellic acid. Could you comment on those studies as far as you know of them?
Yes, as I just said, and it's a little bit more complicated than that. It induces and that's it. In some cases, we don't know in citrus yet. That's the problem. It has been done in several other species. In some cases, is depending on the dosage, meaning that lower dosages induce an immune response, higher doses inhibit that immune response. And that is something that is common in many hormones, but in the case of brassinosteroids, has been really well studied in Arabidopsis, for instance, has been studied. So we still don't know. The rate that I've shown here appears to work for us, but I don't know if going lower would be better or going higher. We haven't seen an increase. Going lower could be better. We didn't see that in mature trees, for instance. Okay, in mature trees, what I just said regarding to rosmite was in mature trees at that doses of 6.2 ounces per 100 gallons. Below that doses, we didn't see any effect on rosmite in the surface of the fruit, because I didn't say that, but we were assessing the effect on the surface of the fruit. So we were looking into the area of rosmite there. So we got a decrease in the area of rosmite after brassinosteroid treatment. Now, that's what we know. What we are trying to do now is do the treat. Well, we already did the treatments and we have the samples, and look into jasmonic acid production, gene expression of jasmonic acid, because actually we have those genes identified in citrus, so we can know. And also to look downstream that, because downstream jasmonic acid is very interesting, is one of these volatiles that I just mentioned. A family of those volatiles come from that. So if we are seeing this repellent effect, probably is because of that. I say probably, and this is a hypothesis, we don't know yet, but probably this is what the effect that we are seeing is because of that. I don't know if I answered or.
Any other question from the center? Any comments? Wait.
Yeah.
I can talk loud.
We have people on this one.
Yeah.
All of this seems very interesting. And I know it's restricted to just getting young, healthy trees a good head start. What is the next step as far as maintaining the young trees you now have, which is a huge investment, and then bringing them forward into having maybe at least 10, 15 years of production for a tree? Is there another set of parameters that you would use to help fight
Yes.
infection and increase the immune effect that the
Yes.
trees now have a starting of?
That's a great question, and that's our goal. And we are learning as we go, because we started with these trees 5 years ago. Actually, I started in this position 6 years ago. So we are learning as we go. And what we are learning is that we should start to have a new set of tools in our toolbox that will probably require IPCs at the beginning, then probably some bridge, and I would say bridge treatment with brassinosteroids or other plant growth regulators that can work, like brassinosteroids or others. And after that, as the tree starts declining, and we are already in the 6-7 years, we have the next presentation is going to be Dr. Albrecht talking about OTC. Then probably, because the trees have a trunk that is already capable of getting the injections that we'll need, then probably that's the time to treat the trees with OTC and try to get that health in the trees until 10, 15 years. Why not? I don't know. I don't know that. I mean, I'm not an economist. I'm trying to. I know that. I know. I guess that will depend on the yield that we get and the quality that we get for those fruits. And we are still in the second, third.
I was curious, that would be the long-term approach.
Yep. Yes, and as I tell you, we are learning as we go. We don't know. But I mean, if after all this time we get a treatment that works, like for instance what we are seeing with OTC, and we get better quality, and actually we are able to heal the trees to some point, then probably yes. The problem is that and Dr. Albrecht will talk about that later. The problem is that probably in young trees you cannot do that. Has to be older trees, but she will address that. But the thing is that we are trying to put together some tools that were existing or not by separate, to see if it makes sense for growers and for the industry in the long term. That's what all we want.
Any other comments? Oscar.
If I can only get this work, okay, thank you, Alison. Thank you, Fernando, but if I may just comment what
Sure.
was just asked. So short answer to long-term solution, we don't have it, by the way. This is what we have so far, short-term solutions for the HLB, unfortunately. That's why our industry is declining. Ultimate solution would be more tolerant and resistant varieties that we don't have it yet, but we are all working. So you will hear from multiple scientists that everybody is tackling this HLB situation from different angle. What Fernando is doing is actually keeping the young resets a little more productive until they eventually get infected with HLB. So the ultimate solution will be more tolerant and resistant varieties, unfortunately. I have a question, if I may, Fernando.
Yep.
You showed extremely nice result with the immune response that the Brassica steroids are giving after 5 days of application. Were you able to follow how long that immune response lasted?
Not yet. We are doing that now.
Okay.
So we have the samples, and eventually we will have those data.
That will be very interesting, actually. Thank you.
Any question or comment from the Zoom group?
I see one over there.
Go ahead.
Chat. Yes, steroid application. Are you doing studies on mature plants? Yes, as I said, yes. Yes, we are doing that. And what we see is less impressive than in younger plants. But for instance, the effect that I just mentioned on rose mite, that was on mature plants because they were fruit-bearing trees. So in that case, yes. The thing is that in mature plants, the immunity is not as strong as in younger plants. So it's normal to see less effects or not that spectacular than in smaller plants. And actually, we haven't seen after 5-6-year-old trees, we haven't seen anything that we can actually measure or report that is different. But in younger plants, yes.
Any other question? No one. Thank you, Dr. Alferez.
So good morning, everyone, and thank you for being here in person. It's nice to have presentations available by Zoom, but I feel it's much nicer to have in-person meetings. So I'm going to talk about what you need to know when using trunk injection to manage HLB. You heard Dr. Alferez's presentation. So the IPCs and the steroids give you several years of productivity and tree health. And of course, the trees become reinfected. And what can you do then? So I have my presentation divided into 5 parts. So I will begin with some of the principles of injection, then what results we can expect, some of the concerns surrounding oxytetracycline injection. Then I'll talk a little bit about the label, and I will conclude with some best practices. So let's begin with some of the principles of injection. We're all very familiar with HLB. The only thing that I want to highlight or emphasize here is that HLB is really a vascular disease, a systemic disease. You can see the box here on the top. It is the bacteria. These are the little rod-shaped structures. They are sitting inside phloem cells. So it's a systemic disease, which makes it so difficult to cure. So we can't really spray our way out because it's everywhere in the tree. So what is the vascular system of plants? Well, it consists of 2 parts. One part is the xylem. The xylem consists of xylem vessels, which are dead lignified tubes through which water and nutrients are being transported. The transport is passive, and the driving force is transpiration. And as you probably know, transpiration is the process of water evaporating on the leaf surface through the stomata. And this evaporation then in turn pulls the water from the trunk, from the stem and the roots upwards into the leaf. And so it moves water and the nutrients passively. And the transport is, like I said, from the bottom to the top, so it's unidirectional. In contrast, the phloem is the part of the vascular system, so it's a live tissue. And this is where our pathogen, I have it abbreviated here as CELAS, is located. In the phloem, we usually have a transport of carbohydrates, namely sucrose, coming from the photosynthesizing leaves, and some other organic compounds can be transported. The transport is an active process, and it can be in different direction, but it's usually from source leaves, from source tissue, like photosynthesizing leaves, to sink tissues, like, for example, roots or developing fruit or other developing leaves, for example. So let's look at the anatomy of a tree. So on the left, you can see the little pie-shaped or wedge-shaped thing taken out of a tree trunk, and you can see these green globules here. These are the xylem vessels, and if you look on the side here, so you can see that they're long tubes, and again, these are hollow tubes that are lignified, so they're dead cells, but it's interspersed by, do we have any, don't know if I have any pointer here. Anyway, so you see the rays here, these stripes through this wedge. These are living cells. Actually, these are parenchyma cells or ray parenchyma, or also these seasonal growth rings. These are made up of living cells. And then we have the yellow here that is right underneath the bark of the trunk. This is the phloem. So it's the living tissue again, and this is where our pathogen is located. So what exactly is trunk injection? Well, it is the targeted or vascular delivery of agrochemicals or crop protection materials, whatever you want to call it, into the stem or the trunk of a woody plant, and as an alternative to spraying or soil trenching. Some people also call it endotherapy, and some people don't like that term. So the injection occurs into the xylem or the wood of a tree trunk, and from there the materials are then distributed throughout the plant with a transpiration stream. And this is what makes it so powerful, because it's a systemic delivery system as opposed to a foliar spraying the trees. So how does it look in a tree? So I'm talking about these syringes here that you see in the photograph on the right, and a little bit later too. So this was our way of injecting trees. And on the left, you see a cross section through a young citrus stem that we had injected. So that's the injection scar. So after injection, then whatever we inject gets shot into the xylem or the wood of the tree. And again, our phloem is just a very narrow tissue right underneath the bark. And then as whatever we inject moves upwards into the tree, it's assumed that it can also move radially from the xylem into the phloem in order to be effective against the bacterium that is in the phloem. So before we did actual field studies, we wanted to see what really is going on in citrus trees. So we did some simple dye studies. Here, the dye we used is acid fusion. It's very pink, as you can see, and we injected a fairly young greenhouse tree just to study the simple principles of injection. And here, the picture on the top in the middle is our injection site. You see this big plop of pink dye. If you look on the left, this is the dye that leaks downwards. That's actually a cross section that we cut just below the injection site. So you can see a bunch of plops, which means that the gravity will push some of what we inject also downwards. And as we go up into the tree here on the right, you see some more plops. And as we go further up into the tree, these plops will finally emerge into some spiral pattern. And so you can see basically it's kind of like growth rings, and these globular dots are indicating that the dye is moving within the xylem vessels. And so this is a good thing. The spiral pattern of ascent is what most trees have, but there are some exceptions. And it also means that if we inject on one side, so in theory, at least we can have whatever we inject move into other sides of the tree. So some trees are sectorialized. That means if you inject on the right side of the tree, it will only move on this side. But we have a spiral pattern of ascent, so which is very good. So, and as we move further up into the tree, the dye or whatever we inject then continues to move upwards and into the outer rings into the outer growth rings until it's finally in the sapwood. And then the most recent growth ring or the sapwood is where actually most things are actively transported. And as we move further up into the smaller branches, you can see here the dye finally concentrates. So trunk injection is nothing new. For many centuries, people have played around with it. Arabic horticulturists had put dyes and spices into wounds of trees to make their orchards smell pretty and look more colorful. But there's also, I copied it because I thought it was kind of amusing, some anonymous entry here from 1602, how sour fruits be made sweet. So in this case, they drilled a hole into the tree and put honey in it to make the fruit sweeter. And then on the right, you can see how the worms are to be killed if they've already been grown into the tree. And by worms, they're likely meaning insect larvae that bore in the wood of the tree. So they're drilling a hole in here and take pepper, laurel, I guess it's bay, and incense and mingle it well together with some good wine and then pour it down into the hole of the tree. So I do not recommend it, but I just want to reiterate that people have been playing around with this for many centuries. And there also have been some more scientific research in the 18th centuries, and this were actually worldwide. So what are the modern areas of use of trunk injection? Well, it's mainly used in forest and ornamental trees and large woody shrubs, and also on palms in residential and commercial landscapes. But it's also used on some crop-bearing agricultural crops, for example, avocado, peach, or pear. And in Florida, 20% of commercial avocado production is actually treated by trunk injection on the commercial level for laurel wilt. So the targets are pretty much any insect that feeds on the leaves or other parts of the trees, including wood-boring larvae, also wood-boring nematodes, fungi, phytophthora, also bacteria and phytoplasmas. And phytoplasmas are the cause of the lethal yellowing or bronzing in our palm trees. And they actually have been using successfully oxytetracycline injections to prevent the trees from declining from phytoplasmas. So when I started on this research, we checked online, and there were actually a lot of different injection technologies on the market. I was really surprised about that. So here are just a few of them, but you can see that most of them are drill-based, and few are non-drill or needle-based, but all use relatively large injection holes. And you can also see that the trees that they're using are fairly large, not like our citrus trees. The injection device that, and I'll talk about the label later, that's the injection device that the company TJ Bio has developed. So that is just a picture of how it looks. Some of you may have seen it already. Nothing fancy, but it's effective. You just have a valve and you have a balloon-like tubular structure that adds some pressure, and I'll talk about that later. So we also did some preliminary studies regarding the injection pressure. Is it better to inject things very quickly at high pressure? Is it better to have kind of like an IV procedure where we rely on passive infusion? Well, we did not really want to continue using high pressure injection because it's very invasive. It causes a lot of damage to a small tree trunk. And we found that low or no pressure infusion is not very effective for delivering large volumes of liquid materials. It works well in the greenhouse for testing therapeutics, but in the field, it's not very practical. So we decided in the end to use these syringes that are delivering things at a pressure of 25 to 30 PSI. And this is what we have been using moving forward. Of course, the new device that is recommended by TJ Biotech is this that I just showed you. It has pretty much the same pressure. And so even though they look a little bit different, the basic principles are the same. So we also did some studies on transpiration rate. If you remember, I explained what transpiration is, and that is driving the process of moving things upward in a tree. So in this figure here, we measured the transpiration rate indirectly by measuring the stomatal conductance of the leaves. And we did this in July, September, etc., until May. And you can see between July and November, the trees have a fairly high transpiration rate or stomatal conductance. And it drops suddenly here in March. Why does it drop? Well, it's because that's the time of the year when the trees were flushing. So they had a lot of new leaves, not fully expanded leaves, which obviously then hinders makes the transpiration not very effective. So the transpiration rate of the tree determines the speed of uptake and the distribution of injected materials. And it also depends on the weather and the season, time of the day, etc., and the physiological state of the tree. Then there's the question of rootstock versus scion injection. So we had started out by, most of our research has involved injection into the scion. It was simply because it was more consistent. We pretty much have sweet oranges, mostly in Florida, and we have numerous different rootstocks. So we assumed there will be some variability depending on the rootstock. And also, if you inject into the rootstock, they have a closer proximity to the soil. So we figured you would have a higher risk of secondary infections of our injection wound with soil-borne pathogens like Phytophthora. Anyway, we did some preliminary experiments. However, you can see here the figure. We have the red bars are for injection into the scion and the green bar for the rootstock. And again, you can see we did injection from July till May. So if you remember the previous slide I showed you about the transpiration rate is pretty much fairly correlated with the relative uptake rate that we measured here. We had the lowest uptake rate in March, regardless of whether we inject into the rootstock or the scion, which is because that's where we had the lowest transpiration rate. But you do see some differences here. In this experiment, overall injection into the scion led to a faster rate of uptake, but overall both methodologies worked. So what results can we expect when we do trunk injections? So one of our first field studies that we conducted were conducted here at our center at SWFREC. We used 5-year-old Valencia trees that were grafted on Kuharski rootstock. It was the COVID year, so we wanted to start out with our injections in spring or in April, but then the center was closed. So our first injection we performed in October that year, and then we did another injection in April of the following year. We used some commercial, some formulations that were available as injectable formulations. So we used imidacloprid from a company called Zytec. Again, it's an injectable formulation. And then we purchased oxytetracycline. It's called Arbor OTC from Arborjet. It's also a formulation that is made for injection. And we used an active ingredient rate per tree of 0.79 grams. We compared that with water injection or no injection. And as I mentioned, we use these syringes or chemjets, what they're called. They are basically spring-loaded syringes. So they're injecting at a medium pressure. And we're usually using 2 injectors on opposite sides of a tree, and each holds a volume of 20 milliliter. The trees that we used in this study were 25 to 3 inches in diameter that's above the craft union, about where we injected. So let's talk about the imidacloprid first. We, together with the help of our entomologist here at our center, Dr. Javad Qureshi, we bagged some of the trees with these little mesh bags. And we used lab-reared colonies of psyllids to avoid that the psyllids already are resistant to imidacloprid. So the trees were bagged after we injected. And here are some of the data. On the left figure, you can see the psyllid mortality that we measured 1 to 2 weeks after injection. Imidacloprid is indicated by the green bars and water by the red bars. So you can see that imidacloprid injection led to a 50 to 70% mortality compared with no or water injection, which was less than 20% mortality. So definitely a significant effect. But when we look on the right, 2 months after injection, we do have a statistically significant difference, but it's only, it's probably biologically irrelevant because we have a mortality of less than 20%, which is likely not being effective. I mean, it is effective initially, but as you know, the psyllids are abundant and they will come back. So imidacloprid works, but it's not long lasting. However, this was just a short study and we used just 1 rate, so it may be worthwhile to do some more studies on this formulation. Oops, I forgot to highlight it, yeah. So what about oxytetracycline? So obviously fruit drop is one of our major problems here with HLB. And on the left, you can see the fruit drop. Oxytetracycline is indicated by the blue bar. And I was extremely surprised, but we were able with this injection that we measured a reduction in fruit drop. We reduced the fruit drop to less than 25% compared with the other treatments, which were like 70% to 80% of fruit drop. So that was pretty impressive. And in the year afterwards, this effect was also observed. Again, we see fairly similar results. So what about the effect on the yield? Obviously, if we have less fruit drop, we will have more yield, and that is pretty much confirmed in these figures here. We have increased the yield from whatever, less than 2 to 5 kilograms of fruit per tree to almost 10 kilograms in the first year, and similarly in the second year. But a picture is worth more than words. So this is pretty much how it looked in our fruit bags. So I was quite surprised that we had such a tremendous effect because I didn't expect this to happen. Also, fruit quality is important. I don't know, colors don't come across very well here, but on the left, you see a figure that measures the color ratio with a colorimeter. And you can see the oxytetracycline has a much higher color ratio, which indicates that the fruit are more orange as opposed to be green, which hopefully you see on the photographs here. So for the first time, we actually had orange fruit in our Valencia trees. Bricks, obviously also important in addition to the yield. If you have more soluble solids, you'll get more money. So we found that the oxytetracycline injection increased the bricks. You can see here it's over 10, and I think they were harvested in February. So that's pretty good. And in the second year also that's a bricks acid ratio of about 11 compared with the other treatments, which were less than 8. Fruit size, there was something interesting that we found in the first year after injection. You see we had an October injection. So we injected after the fruit had already going through their developmental phase and were already fully enlarged by the time we injected. So we did not have any effect on fruit size. But if you look on the right figure, that was after the April injection, you can see there was actually a dramatic increase in size. The size almost doubled. And if you look at the fruit, my student said it was almost comical. So we did an experiment afterwards where we did injections either in October or in April or no injection, and this is what we found. So you see the October injection, the fruit are much more orange compared with the no injection, but the April injection are just huge. And we had to harvest in February because we wanted to get data and not lose all the other fruit to fruit drop. So if we would have waited till later in the year, I guarantee you that these fruits would have matured and probably had a fairly high fruit quality. We are so used to now doing the harvest in February, so maybe the injections will help us to get back into our old rhythm of harvesting Valencia fruit much later in the season. So, of course, there's a big concern regarding the residual levels of oxytetracycline in the fruit. So we had done some initial studies. We were told it is best to measure the residues in whole fruit per regulation. So not in the juice or not in the peel. So just mush them all, to mix them all together. And so we used a service from a accredited laboratory from USDA that is in Gaston in North Carolina. So that's known for food measures, adulteration of food products. So it's a highly sensitive test that they used. And you can see on the x-axis here, the time after injection, we measured 6 hours to 2 days, a week, and up to 300 days after injection. And you can see that after the injection, 30 days after injection, the residue levels drop pretty drastically to very, very low levels. So we have almost no residues in the mature fruit. The currently allowed residue level is 0.01 ppm, that's 1 part per billion. So it's very low, but we managed to get there. And also, if you remember when we had the section 18 label for foliar application of oxytetracycline, our allowed residue levels were actually 0.4 ppm, so 40 times higher. But I guess it was not renewed, so this is why the regulatory agencies went back to the default, which is 0.01 ppm. And I may also mention that in peach and pear production, where they're using oxytetracycline foliar, their allowed residue level is 0.35 ppm, so 35 times higher than this. So I think these data are pretty encouraging regarding the residue levels in the fruit. And if we measure it in the juice, you probably even have less. So we also measured the oxytetracycline efficiency in terms of what does it do to the tree, especially to flushing and flowering. So on the top, you see the different stages of flushing. We measured it in 4 stages here, from 0 to no flush, to feather flush, to flush elongation, leaf expansion, and leaf hardening. And this complicated figure here on the left with all these peaks is not as complicated as it looks. So we measured over time the proportion of these different flushing stages. And just if you look at the pink, at the reddish pink peak, that was the peak for oxytetracycline, you can see that oxytetracycline induces a much better synchronization of the flush. So whereas you can see all the green and the blue for no injection or imidacloprid, they're all over the place. The leaves never really go through the full flushing and elongation and leaf hardening stage. So oxytetracycline injection synchronizes the flushing. And I didn't show the data, but also it did the same for flowering, which is really good because if we can do all these different management strategies, from IPCs to steroids to maybe trunk injection, we can basically keep the physiology of the tree as to kind of where it was pre-HLB, and then we can also focus our psyllid control sprays to the main time of flushing. Like trees are flushing, they're probably gonna mainly in the spring is the main flush. So if we focus our sprays there, of course they're going to flush a little bit throughout the summer. But if we can make some targeted psyllid control, so that would be another help maybe with keeping the trees healthy longer. So this is what I was talking about, oxytetracycline. See, the leaves are fully expanded and much larger than what we see here in the control leaves. And we're all too familiar with these yellow chlorotic leaves that are caused by HLB. So we also measured the HLB foliar symptoms and the canopy density and the foliar symptoms. It's hard to see the bar because the data were so tight. You can see the oxytetracycline reduced the foliar symptoms and also increased the canopy density significantly compared with the other treatments. What were the effects on growth? So here you see three figures. We measured the trunk, the rootstock trunk circumference on the left, scion circumference, and then the increase in height. And measuring within 1 year an increase in rootstock and trunk diameter circumference is pretty impressive. But you can see that in all cases, the only oxytetracycline actually led to a much improved trunk diameter increase and also tree height. This is a picture of a set of trees on the left. It was 12 months after the first injection. So it's pretty obvious which tree was injected with what. So these results were pretty impressive. So these were fairly young trees, and they're fairly strongly affected by HLB. What happens when we go in a commercial location and we inject mature Valencia trees? So this is a photo from one of the studies that we did. We used 8-year-old Valencia trees on Carrizo. Here we did treatments that we injected either in April or in October. So no double injections, only one injection in April or in October. And we used the same rate that we used for the smaller trees, so 0.79 gram per tree. So what did we find? Here we see the graphs for fruit drop, yield, and fruit size. This time the blue bars, or I guess it looks purple here, for the control, and the April injection is green and the October injection is red. You can see in all cases, April and October injections significantly reduced the fruit drop. However, in this case, the fruit drop for the control was not that bad. It was less than 30%. But it also increased the yield, especially in this experiment when we injected late in the year in October. We had a more significant effect on yield. And as I mentioned before, we see again this significant effect on the fruit size, especially when we inject in April at the beginning of the fruit developmental phase. You can see a larger increase in fruit size compared with the October injection, although there was still a difference compared with the control. The results were not quite as impressive for the young trees, but that is to be expected. But definitely a significant increase in profitability. So what are the concerns? So obviously, if we drill a hole into the tree and then we inject something, we're causing injury to the tree. So does it matter? Yeah, I'm sure it does, but I want to show you some pictures of how it looks. Here on the left, this is what happens after water injection. You see the hole is fairly small and it's also closed. So the wound is closed fairly quickly after we inject water. The three photos here on the right are what different things that we see when we inject oxytetracycline. So it's pretty clear that oxytetracycline delays the wound closure and also increases the wound size. There seem to be very random effects. Sometimes we see, like on the right, this bark cracking is we see often, but then sometimes in the middle, there's like what we call wound wood formation, and or on the left, bark cracking. But the wound usually stays open during the initial months after injection. So it takes a little bit longer. But I also, I'm not showing data, but we have submitted a paper on this where we compared like outside with inside damage. If it looks ugly on the outside, it doesn't necessarily look uglier on the inside too. So sometimes it looks worse on the outside, but the inside is just not any different than wounds that look better. So let's talk a little bit about the concept of coded. Coded stands for compartmentalization of damage or decay in trees. It was developed in 1977 by two researchers. I think they were arborists, to explain what trees do when they are wounded. That not necessarily refers to injection, but it can be any trunk injury from insects or whatever, just physical injury. So I just want to show that briefly to explain you what's going on inside the trunk. So based on this coded model, it depicts the tree as a chambered organism and where there are walls formed after an injury. And here you see three walls. Wall one is the wall that is indicated by the beige. So that's the wall that is formed after injection in the up and down direction. So in the axial direction, that's determined pretty much by the length of the xylem vessels. Wall two, indicated here by red, is the wall that is formed from the outside to the inside, so towards the center of the trunk. So that's in the radial direction. And then wall three, indicated by gray here, is the wall that's formed in the tangential direction, so from left to right. And wall 1 is the weakest, and wall 3 is a much stronger. So how does it look inside a citrus tree? So here you see a cross section on the left and the longitudinal section on the right after water injection. So you can also see that citrus trees are able to compartmentalize wounds very efficiently, at least if we inject water. So here's our wall 1 in the up and down direction. So not you can see there is some discoloration going on. Wall 2 is pretty much determined by how deep our drill is put into the tree, so very effective. And if we look at wall 3 here in the tangential direction, again, is pretty much determined by the size of the drill bit when we drill the hole. So very effective wall 3. There's another wall called wall 4, or the barrier zone, that is formed after the wounding, and it's the strongest wall, and that is the wall that is formed between the old wood and the new wood. So once the tree grows again and puts new sapwood in a new growth cycle on, those growth rings are separated by a wall to prevent the potential decay inside the trunk to move into the new growth. So trees are actually very effective at doing that, and it's not really called a wound healing, it's more a compartmentalization. And these walls are formed by the living cells in the tree, those ray parenchyma cells, or these parenchyma cells in general. So what happens when we inject chemicals? So on the left, you see a cross section of a trunk when we injected water. So you basically just see the wound site here. Imidacloprid in the middle, you can see a plop of gray here in the center of the trunk. And if you look at oxytetracycline, this plop is fairly large. So it doesn't look pretty. And initially, I thought this discoloration may be associated with the pH of the solution too, because oxytetracycline, as you know, has to be acidified, and the pH was 2, so very acidic. But imidacloprid formulation, we measured the pH as a pH of 4, so less acidic, and you can see it's not as strong. But we did just some recent experiments where we just injected muriatic acid into a tree, and we really didn't see that brown. So one interpretation is too that the discoloration is simply due because of the coloration of the formulation itself. So it's a very dark brown solution. So how does it look in the longitudinal direction? So also not very pretty. Remember, wall 1, or the compartmentalization in the axial up and down direction, is the weakest, the least effective. So water on the left looks pretty good. Imidacloprid in the middle, and oxytetracycline here on the right. And again, oxytetracycline is darker in coloration than imidacloprid, so some of it may have to do with it. But in any case, it is likely. Oh, I have actually some wood here that you can hand pass around. So even though it's discolored, it's likely that these vessels here are not functional anymore. But however, it's not a structural decay, so they are structurally intact. So it's not that the tree is like crumbling apart during the hurricane or anything. So it is pretty strong. Also here, this is what I was talking about, this new wall, the barrier zone, or wall 4, that is formed in the new growth cycle. And you can see the sapwood here, the new sapwood is, after a year of injection, is perfectly healthy. And in fact, it's much thicker after oxytetracycline injection than after water or imidacloprid injection. So that is pretty much what we measured in terms of a trunk diameter increase. So something else I want to point out. So as you saw that here really nice on the imidacloprid injected trunk. You can see it's kind of narrower at the right, and then it goes much larger on the left. This is because this is the older wood. So if we inject into the older wood is metabolically not as active. So it has a much harder time to compartmentalize or heal the wound. So this is why there is much damage. So for comparison here, if we inject not as deep, the wood is much metabolically much more active. So the wound is more efficiently compartmentalized. So what does that mean? It's like, when you do injections, try to inject as shallow as possible. So don't just drill the hole as deep as you can into the tree to make it work better. You're causing more damage. So the shallower the injection, the better. And that goes for pruning. And so this is just a basic principle for tree wound compartmentalization. So what we also measured is some seasonality with trunk injection. So here you see some data from the percent. This is for the external wound. We measured the percentage of fully closed wounds at 4, 8, and 12 months after injection. And we did that after June injection, after October injection. Here you see the data for water. When we inject water in June, 4 months after injection, 100% of all wounds are closed. When we inject water in October, again, the tree is not as metabolically active. We only have 25% of the wounds closed, but after 12 months, all the wounds are completely closed. For comparison, when we inject oxytetracycline in June, it takes 12 months to have over 80% of the wounds closed. And if we inject into October, it takes 12 months to have a little bit more than half of the wounds closed. So this is what the photos I showed you. So oxytetracycline delays the wound closure, and if we inject into the fall, it's even more delayed. We also tried some different wound treatments. So here on the top left, you see some pruning sealant that we purchased to put on the wound. It's tar-based, so it did not look very pretty. We found that it, in fact, interfered with the wound healing and caused more damage. So I do not recommend to do anything like that. And it's everything I read in different arborist or forestry papers. So the tree is very good at compartmentalizing. Just leave the wound alone. Don't do anything. Don't put any stoppers or things in there. I know the avocado industry who's doing it. I mean, avocado trees are bigger, but they inject bigger holes and they're using these golf tees to plug the wound. Don't do that. Just leave the wound as is. You know, we also put some Ridomil on some of the wounds for potential, you know, Phytophthora infection. We found that the Ridomil didn't have a negative effect, but it also made the wound in some cases a little bit larger internally, as you can see here. And we also compared rootstock with scion wound. Here's a little figure regarding that, and we found that the rootstock actually, in combination with the tar, had a larger wound size than when we injected into the scion. So there are some interactions between wound treatment and also in the rootstock. But we're still continuing some of these studies. But I guess to summarize, just don't do anything with the wound. So we talked about oxytetracycline and imidacloprid. So of course, maybe it's not all that desirable to have an antibiotic put into the tree. So I just want to assure you that there are many researchers working on other therapies, whether they are folia or otherwise systemically delivered or by trunk injection. I have a project going on with some colleagues in Texas that have developed a nice system called the hairy root system to screen libraries of random compounds with potential efficacy against bacteria, especially CELAS. And some of these compounds, some of the best ones that were discovered in this program, we're testing right now in our field. Here are the chemical structures from 3 of them. And I'm still waiting for the data because we just started the injections this year. I just got the data from the collaborators in Texas who are using grapefruit. And they surprisingly had some pretty impressive increase in fruit yield with some of these compounds and even when compared with oxytetracycline. So that's exciting, and it is funded by USDA NIFA grant. So again, there is a lot of research still being done for other potential therapies. So let's talk a little bit about the label. So as you probably know, in October, a special local need label was approved, a section 24C that now allows the use of oxytetracycline in Florida for citrus. And just, I guess, the other week, it was finalized for use after the 90 days time for EPA to be against it. So it is now official. This is the label for Remedium. It is pretty much just 95% oxytetracycline hydrochloride. And I have some handouts. If you look at the table back there, you can take some of these handouts out. Dr. Dudney, Dr. Batuman, and I worked together to put some kind of like advice and guidance. So it's an informational sheet on how to use the oxytetracycline injection and some of the concerns, etc. So you can look at it. We also have a muriatic acid safety sheet because you will need muriatic acid to acidify your solution. Okay, the label says you can use it on bearing citrus and you can use it on non-bearing citrus. The volume and the actual rate that you inject depends on the trunk diameter of the tree. And for the non-bearing trees, they recommend that you do not do it when the tree has a trunk diameter less than 1 1/4 of the inches. But they say it's okay to do it 1 1/4 to 2 1/4 inches. However, I advise, even though it's allowed on the label, I know, but I advise strongly against injecting tree with a trunk diameter of less than 2.5 inches, and especially not injecting more than once. According to the label, you can inject twice in the year non-bearing trees. If you do that, I would think that you may kill your tree just from the injection. So, but it's up to you. So, like I said, the recommended rates will depend on the tree size. For like a mature tree with a trunk diameter of 4 to 6 inches, I think that refers to the just above the craft union, will receive at the maximum label rate 1.1 gram. And remember, I used 0.79 grams, so we were right in between those rates. And bigger trees, the maximum rate per tree is 1.65 grams. And again, non-bearing trees here, the label rate is much lower, so I'm not sure. Again, I don't recommend to do this. So what are the costs? So these are the costs with that label that was approved for the remedium. This is the cost per acre, varies depending on the label rate, the trunk on the rate and the trunk diameter, but at the maximum rate, it's anywhere from $200 to $400, and that is just for the material. It doesn't include labor, and it doesn't include the injectors. However, yesterday afternoon, another label was approved. I don't know how many of you have heard that. So it's from a different company. I won't say too much about it, but I know the pricing will be different. So that is that. I've talked to some growers regarding the labor. I've heard something from some people have figured out a system to do it $1 per tree. But just yesterday, I talked to someone else. Their team of people has calculated they can get away with 50 to 53 cents per tree. So I guess growers are inventive, and if you have to make it work, you figure out a way to make it work. So I want to end with just some best practices based on the information I talked about today. That's my advice. Inject after the harvest and after the main flowering period, because we really don't know anything about the pollinators. So if you inject after the main flowering, you prevent exposure to pollinators. I know a lot of people are already injecting, but I would advise to wait. The uptake and distribution of injected materials is driven by transpiration, as I explained before. Therefore, I inject when the leaves are fully expanded, not during the leaf flush. So you can ensure efficient uptake and distribution of your injected materials. And if possible, inject during mid to late morning. This is when the trees are most actively transpiring. The injections in the afternoon are likely less effective simply because the tree closes the stomata and doesn't transpire anymore. And you also may increase your risk for phytotoxicity when it's really hot. So that can potentially happen. Regarding phytotoxicity, it's also best to avoid injecting during the hottest time of the season. There have been some reports that when injections were conducted in August, that it led to some phytotoxicity that was manifested as leaf bronzing. I know these trees have recovered. I have never seen it. We did some injections in August. I haven't observed it, but I know that it has been observed by some other people who have been experimenting. When you inject, make sure that your equipment is in good shape. Use a sharp thread point drill bit for drilling the hole to minimize the injury. As I mentioned before, drill the hole no deeper than necessary, because the deeper the drill bit, the more damage occurs into the tree. Do not use any wound sealants or plugs to plug or to seal the wounds, because they inhibit the ability of the tree to heal on its own. And then remember that the trees will become or are very likely to become reinfected. So oxytetracycline injections are no replacement for psyllid control. You will need to control the vector if you want to make the most out of it. Although the label allows it, I advise against injecting tree with a trunk diameter that's less than 2.5 inches. And I would also inject trees only once. And the label for mature trees is only for injecting once anyway. And of course, you have to leave a pre-harvest interval of at least 180 days. And lastly, follow label directions. The label is the law. We have published a bunch of things. Some of them are extension materials. Some of them are research publications. If you're interested in reading more of our results and some general information on trunk injection, you can contact me. I can send you copies of these publications. And finally, I want to thank the funding agencies, USDA, NIFA. All the research that was done that I presented today mostly was done under a grant that we received together with Dr. Batuman, Dr. Alferez, Dr. Ampansidis, and other collaborators from other centers and institutions. And I have another grant going on, like I mentioned, with the Texas people to look at other therapies. And CRDF is also funding some ongoing research. I've only talked about one mature field trial. I have a bunch of trials going on right now. Unfortunately, I don't have the data yet because we're still waiting for the yield. Some of these, all of these trials are with commercial collaborator and mostly sweet orange trials, but we're having trials together with Envio. There's also another company, with TJ Biotech, and there's another company with Envio that is likely to put forth a label maybe this year. So there's a lot of stuff going on, and I'll keep you informed about some of the new findings. And of course, I want to thank the grower collaborator, my awesome team here at SWFREC, and special thanks also to my PhD student, Dr. Lee Archer, who conducted most of the research that I'm presenting today. And that concludes my presentation. I'll be happy to answer any questions that you may have.
Any question from here?
Dr. Albrecht, I noticed that the trial volume that was injected was 20 mil. However, the smallest
40 because we divided it into,
Okay.
yeah.
smallest label rate is 25. It looked like on the label.
Yeah, we did all this stuff before we knew that TJ Bio is doing their own studies and before we had any idea of the label. So that, like I said, we looked around what is available on the market. We found a commercially available formulation that actually had a label for citrus at the time, the Arbor OTC, but only for non-bearing citrus. And then the chemjets have the maximum volume of 20 mil. And so we just used that. And this is maybe why this discoloration was fairly dark, because it was much more concentrated. So if you calculate in ppm, we were over 19,000 ppm, and the maximum rate for on the label is was it 11,000 ppm. So.
And at that volume at 25 psi in the syringe injector, there must be tremendous variability. But how long did it take?
Anywhere from 5 minutes to 20 minutes. So we usually, we'd usually have a team of 2 people. One drills the hole, next puts it in. By the time we're at the end of the row, on the same day, we take the chemjets back and we can reuse them, we clean them and stuff. And yeah, sometimes we go back the next day to pick them up. It depends. It's really depends on the environmental conditions, time of the day, and blah, blah. You probably want to make sure the trees are well watered before so that they're metabolically active. And even sometimes you have 2 injectors in the same tree. One will be empty in 5 minutes and the other one still like half an hour later or something. So I guess it's a hit or miss what vessel you hit, when you drill the hole. Just random, I guess.
Measure throughout the tree, how quickly you could pick up the OTC, say if you injected it in the bottom before you picked it up at the top?
We did, yeah, I have so many more data. I didn't show all these data. We did some where we measured the residue levels of OTC, and it was the highest residue levels were 72 hours after injection. So like 3 days or two, so it moves. I mean, that was the earliest I think we did. So I mean, you can, it's a lot of work if you do it. All that. So it goes up fairly quickly, but I actually deleted these figures because I didn't want to drawn on for longer. So we measured residue levels in the leaves, in the bark, and in the roots from like weeks to 300 days or something, or 180 days after injection. And when there was one experiment was interesting. We injected in October, and we actually measured an increase in the residue levels in the leaves towards spring, which would make you believe that it's maybe stored somewhere in the trunk. And as the tree becomes metabolically active in spring, it moves it up, which would be kind of a good thing. But it was one experiment, so I don't want to draw too many conclusions.
Well, that was kind of my next question. If you had a, you know, that your data was kind of as far as early or late.
I'm initiating some experiments, because what we found in these studies that the October injection actually had a better effect on the yield, maybe because it was late, but against all like wounding, you know, results and what is known in the literature, injection in fall maybe causes more harm to the tree. So we have to, I want to do definitely initiating some experiments regarding that.
Well, it certainly is a possibility with Valencias, but not with Hamlins in the fall. But
Yeah, well, that was Valencia, yeah.
You know, it's certainly something to be looked at.
Yeah, and just a moment. And in the bark, we actually measured the highest residue levels, because which makes sense. That's where we inject. And in the roots, we measured very low levels, but our detection method didn't go down. It stopped at 0.1 ppm, so it wasn't very sensitive. But we didn't measure much in the roots at all. I'm sure something goes into the roots, or whether through gravity or if it goes up and then down, I don't know.
So you made a statement about doing bloom or flushing, not to do it. What's your opinion, or why you think there's some activity on bees with the OTC?
I just don't know, because we haven't done it, but I'm assuming it's in the pollen. And there are some reports on the psyllids actually, and I know researchers are looking into that the psyllids, I mean, they have an endosymbiont. For the psyllids, it would be good, because there are some preliminary studies that say, okay, maybe you kill the bacteria. Maybe if we all growers go together and say, now we all inject all trees at once, and then maybe we eliminate the pathogen.
No such.
Very hypothetically. But maybe there are also some good guys in an insect. So maybe, so it's like, we don't know. I'm assuming it's being ingested, and I know that with the bees and the pollinators and neonicotinoids and all that, you know, so just my common sense advice.
You touched on, I guess, just now, but I was wondering if there was any study at all on root mass change, because you showed the yield in the.
Yeah, we're getting geared up to do that. Yeah, we didn't want to stick around there so much in the first year, but I have like I will do that this year. Yeah.
Okay.
So, but my assumption is, you have a healthy canopy, you're gonna have healthy roots. Yeah.
You may have just answered this question, but it looked like when you saw the residue get down to near zero, because it's 0.01 is the current tolerance, it was around 68 days. So wouldn't residue dictate injection time based on a safe buffer?
Yeah, but these are just my data, and they were not replicated data.
Harvest?
Yeah, I don't know what, I mean, I didn't get, the label was from the company that provided data. I don't know what their data was. I'm assuming they had to show some residue levels, you know, so, but, so is your point that you want to inject shortly before the harvest to maximize?
Or just be on the
effect?
safe side, immediately following the previous season's harvest, you inject, because you may not know what.
Yeah.
how
Right now, the
toxins
label,
come off
it's
the medicine.
180 days, so I don't know. So I'm sure there will be more research happening, and yeah.
So Uta, I was just wondering, so you said that you don't want to put it in too far when you drill, right? And thinking also you're trying to get it to the phloem, right? So my question is, on the other side of that, did your team make any determination about it had to be at least this far, like for your kind of syringe, or or the material dribbles out, or it wasn't, effective
Yeah, I
for
should
some
have
other?
brought the ChemJet injectors. That's one reason why we use the ChemJet injectors. I think the smallest drill bit on the label there for their injecting device on TJ Bio, it's was it 13 drill bit? 1364, something? 13, yeah. Ours was 1164, so it's smaller. And that was just perfect for this syringe that we used. And it was maybe like a 1/2 inch that we injected. And the good thing about the syringes, you can, because they're smaller and have a smaller volume, you can just stick them in. You just leave them in the tree. You just put the hole at a slight angle, stick them in. They don't have to lay on the floor, like these injectors, that I guess they're laying on the floor. And then, so you just stick them in and you don't have to stick them in deep. So you're preventing some more damage, especially if you have small trees. 1364 drill bit is pretty big. And yeah, I remember what I wanted to say. We have some study going on, be a while till we know, but we also collected some roots, and I'm working together with our soil microbiologist here at the center, Dr. Strauss, to do some microbiome things, how the oxytetracycline injection, does it cause anything in the root rhizosphere, to change? Yeah, I'm sure that question. I know people always ask about it, so we collected some samples, but it will take a while till we have them all processed and the data analyzed. So.
But as far as just a practical minimum depth, it's just do you need some airspace on past the tip?
Well, it depends on your injecting device. I haven't seen it. I mean, theirs is bigger, so I think they're injecting deeper than I forgot what's on the label, but it's deeper. I think it was 1 inch. What was it? Yeah. So yeah, I mean, it is what it is, So I would inject as when we years ago we did an experiment. We didn't even have the ChemJets. We did someone sent me a handmade device to test something. So it was very funny. We stood there and it's like, how do we inject the tree? So we said, oh, it's in the phloem. So it's drill a really low hole but you can't inject into the phloem. It just doesn't work. You have to inject into the xylem. And the point of injecting into the xylem is anyway that it moves systemically through the tree. The phloem doesn't move like the xylem, not with a transpiration stream. So it's different. That's why I explained that. So you inject into the xylem, because it gets distributed throughout the tree, and then you hope that it gets somehow moved into the phloem, and apparently it does. How exactly it moves from the xylem to the phloem, I don't know. I'm assuming it goes through these ray parenchyma. That's the exchange between the tissues, rather than going up and then somehow down. But I don't know. It's difficult to figure that out, to study it. But I know there are some, I'm not a chemist, but there is an exchange between xylem cells through the parenchyma cells and the phloem cells. I mean, that's like carbohydrates, amino acid, nutrients, micronutrients, things like that.
Any question from the Zoom group? No.
Didn't see anything on the chat. I think they were from the previous talk, right? Yeah, okay.
Question, Ute very nicely showed that is how the xylem vasculature is looking, right? As the many dead channels that is one directional from the root to the top of the trees. So when you are opening the hole, you probably will need to have a little bit of a chamber for the liquid to leave the syringe and hit multiple tubes. So maybe you will be more successful in that way. But very shallow, you will have a risk of, clogging your syringe, and the pressure will push the syringe out of its place. We know this because we are trying to automate this, as you may all know, with Dr. Albrecht and others. Seems like a little chamber for liquid to go in is necessary. So, of course, as Ute was saying, no deep hole needed, but it's some space between the syringe tip and the core of the citrus, a small chamber will need it for liquid to leave the sewage.
So the other company that is probably coming up with a label, they're having their own injection device is quite different than the syringes. Not sure if I'm allowed to talk about it, so I'm not, but it's a very different device that is not it's not round, it's just different. So, but and I think they're working on an automated delivery system for that too. So we'll see. Acrosource. Yesterday at 2:30. apparently. So I heard it in the evening, and so I texted people. It's like, oh yeah, we know since 2:30. It's like, oh God, and I thought I was the first one, but I wasn't. Yes, since yesterday. Yeah, it's the same. The label is the same. It's exactly the same. It's just the cost. I don't know if that's of the costs are official already, but from what I'm hearing, the costs are quite different, more affordable.
The cartridge, we use the cartridge like a bullet shell with a silicone behind it, and I would fill a pouch up with chemical and inject it like you would do a cow syringe. And I would just go from tree to tree and just inject them, whatever.
So you put
CCs
like a
I-
valve in the tree? or something?
Yeah, well, it wasn't a valve, it was like a bullet cartridge. And there's a
That's-
tree
That
saver.
sounds big.
No, it's very small, and we reuse the same one over and over all year long. As many times as we wanted to inject it, but we never took it out and it protected the tree from getting infection.
Yeah, so they say. I mean, we started out, and I haven't shown it with the other system from Albrecht, where they have these little injector. It's like a plastic plug. You drill a hole, you put a little plastic plug in, and through that plastic plug, then you shoot at high pressure. You can only do it at high pressure when you have a plug in there, so otherwise it leaks out and it's sealed tight, and then that plug stays in.
Right.
But it causes so much damage in the tree. I don't like it. I mean, it gets internalized in the tree at
No,
some point.
we didn't
but
have
it's
any
much
problems.
bigger.
We did it
Yeah.
for like, how many
Mm.
years, Jim, did we inject? Was it 3 or 4 years? 3 years.
Well, that's good news. Yeah.
And I used to do the injection of oxytetracycline.
It's a sleeve.
Yeah, maybe.
of material
Yeah.
sleeve?
Brass.
Brass, yeah, brass.
It's a cartridge.
Yeah, from a bullet.
It
It's like
has-
And how do you get it in? Like the easy lens, kind of like you jack
we
it?
drilled the hole.
Oh, you
capped
drill-
it in,
drill.
and then in the back was a silicone backing, and we
Just as a
injected, we
just
injected
seal it, yeah.
oxytetracycline in coconut palms.
Yeah.
That was originally where it was developed, and tree savers are right here in West Palm Beach. And they're the ones that supplied it to us.
Yeah. Oh, okay.
And we used it in the field, and I did some pictures internally. And the tissues, well, of course, we were using a different chemical. The tissue stayed very fresh and healthy, and we reused the same hole over and over and over, the same. cartridge.
You see, there, I'm not sure if that makes sense to me. It's
I don't
using
know.
the same hole, you saw how it looks inside.
Yes, no.
I mean, if you force it in, at high pressure, you can probably get it in somewhere in the tree.
No, there was no
Yeah.
pressure. We just put.
So I'm not sure if it's not like a permanent IV in a tree. I'm not sure. But anyway, I say this, but I also didn't think injection would work. So who am I to judge? So if you have to do it before you
learning.
judge it.
We're all learning.
So, yeah.
It's citrus. You don't.
I did not think it would work, and then we saw those results. I was like, wow. Yeah, so
You know, I brought this oxytetracycline up when Dr. Bouvet, Maurice, I think. When he did his first presentation, and he said his colleague did all kinds of research on it and it didn't work. But
I thought these papers from the 70s, there's a lot of paper where they had IVs and stuff. But
right.
it worked.
Well, he
so-
did it in
couldn't-
South Africa.
Yeah, I know there's a lot.
That publication, in '70s, it worked in South Africa.
I thought that worked in the 70s, but they never did it on, I don't think it was ever taken seriously,
Serious,
you
yeah,
know, and so, but hey, desperation, desperate times, desperate measures.
There you go.
We were just we were completely, we're only down here 4 months. We're cherry farmers from northern Michigan. But actually this drilling of trees, I mean, the maple syrup industry, there's a lot of other
Yeah,
people that
that's
drill
true, they have the
trees and
permanent
they
things,
have
right?
a whole bunch of
Yeah.
things at all these extension
There's a lot of
services up
stuff
there
out there. It's
that
fairly
They know
impressive.
how to drill a little tree and put
Mhm.
it in it and
And
get
I'm
something
sure
in
if
there.
this becomes something that maybe is helpful on a
Maybe.
large scale, I'm sure there will be other technologies or maybe refinements of things, yeah.
Yeah,
And I know, I mean, other countries like France, we were at was it not in Turkey? No, there was a conference in France. Yeah, some people there, I think it was apple trees where they're doing some injection too.
Yeah.
Obviously not for, but not for HIV like disease, but I forgot what it was, yeah.
There is a question on Zoom.
Question on Zoom, do you want to talk? Can they talk?
Yeah, just open it up. They might happen to read.
I open it up? Just click on the chat. You want me to open up?
There we go.
Okay. Based on avocado tree injections, can you comment on annual injections on long-term effects? I haven't seen the avocado trees, but you can, I can, if you send me an email, I can get you in contact with the people who do it. I think they're doing it every 18 months or something, and they have some. Last time I talked to the person, they have developed some conveyor belt mechanism. They sent a group of people in there and they drill the holes, and the next group of people put in some sort of, they inject, and I think they had, yeah, I think it's better if you contact the person directly. And I think they're actually mixing other things in with, it's propiconazole. And I think they're putting phosphide in there too, and some iron. I think they're trying to tank mix into the injection. So, but that's avocado trees. They're much bigger. Oh, I don't know. Yeah, we have done some injections for, I don't know how the effects on avocado trees are. Again, haven't seen those trees. I haven't heard any negative reports, but I'm also not involved in that research. But they're much bigger, have a thicker bark, etc. We have done injections. When did we do the Duncan injections, Oscar? Fernando, how often have we injected those trees? Them twice a year for 2 years. So yeah, maybe we have 2-year data only, but there's some anecdotal evidence that some people have done trunk injections for many years, and they're seeing pre-HLB results. And the trees are apparently still in, if they have done 6 years of injection, maybe Chris has. Well, like I said, if you inject for 6 years and the tree is still in good shape, I wouldn't worry about how it looks on the outside. Obviously, the tree is still functioning. And again, what is your alternative? They're gonna die anyway if you don't do it, right? So you might as well get something out of it.
Separating that
Yeah.
continuity stream from the roots. to
Well, yeah,
to
but
the
so you
atmosphere.
inject into the middle, and then you saw it gets brown, and at least in this area, even though it doesn't disintegrate, it's like unlikely that anything moves through there. It's dysfunctional.
Right,
Yeah.
so if you do this over 10
but
around the
in
tree.
the new year, you're putting a new growth on it.
Mhm.
And if you dip a tree with the roots exposed into a dye, you will see it only moves the dye up in the most outer ring. So the thing that is doing most of the work is the outer sapwood, not the inner brown wood. It's kind of like the hardwood is discolored too. So, but of course, if you inject the new wood again, so I get your point. And yes, of course, it's a concern. And I don't know
I mean,
how the
over time, because you separate those xylem vessels that are going up and down, you're separating them. And if you keep going, I'm not saying it won't work over time, because what you're saying as that secondary.
you go
growth
out
occurs,
more, so
it's
the old
replacing
wood gets
those.
compartmentalized and is really
Right,
irrelevant.
right. So
Yeah,
you still have xylem, but can you overcome the system to where
Yeah.
you're gonna affect
Oh, I'm
translocation?
yeah,
I mean,
that's
we're gonna find out how many trees have blight.
Yeah.
Because you start drilling and try to
I-
inject the
yeah.
stuff, it's not gonna go in a tree.
So I
that
mean,
has
the
blight.
current label recommends 2 years of injection and then no injection. So ideally, you want to inject as few times as possible. So maybe once we get the trees into a good shape, you inject 2 years. And then if you do some proper psyllid control and take care of your tree, maybe you can get away without a tree. I think we had one study where we still had an effect. The trees were infected again. Yeah, you can measure the bacterial levels, but it still had an after effect on the yield. So maybe we only need every 2 years or something. I mean, it's not going to be the only thing we should do in the tree. So don't think, you know, oxytetracycline injection is going to be the only cure or something. You have to do other tree care and management. That includes vector control. You can't grow trees without vector control. We see it in the IPCs. You don't protect them from the vector, they have no chance. You know? So it is still important, and fertilization and whatnot. So, but my hope is that with some sort of integrated management involving vector control, IPCs, and steroids, and maybe an injection every once in a while, maybe you can figure out a way to live with it for a little longer in the hope that we have a resistant variety.
We have one question.
Has there been any work on trunk sprays?
Sprays?
Yeah,
Trunk
actually.
sprays?
I know there was some trunk drench with Alliet, and I think it was pretty positive.
Oh, you mean soil trenches or on the trunk?
No, the trunk drench.
Yeah.
It was labeled for trunk drenching.
Yeah. Oh, paint. Yeah, I've seen some study. I forgot what that was. I know people have asked about that. There is something, and I forgot for what it is, where they have a paint that you know, Dr. No, I think it was something else on other trees. It wasn't in citrus, but I know they have some sort of. Do you know, Jim? Was it written? Yeah. Okay. Yeah, maybe someone will come up with something to paint on the tree. Yeah.
I like to remind that also that recycling is sensitive. It can be bound to silica molecules instantly, so spraying to the trunk will not give any advantage of applying oxytetracycline. Just keep in mind.
I disagree with that. I actually sprayed some on the trunk and had the most beautiful trees you've ever seen for about 3 months. Leaves that big, young trees.
Interesting,
And then
yeah.
it went, and then it went back to greening. But, so I do disagree with that a little bit. But my main question was more maybe up towards Alliet. I know Alliet was labeled for trunk drenches, and a lot of growers that I've talked to said that they feel like we haven't pushed that envelope hard enough, that they've seen some pretty positive results. It was just a curious question if we have done any at the university or not as a.
Well, as you know, there was another permit to use oxytetracycline, and how ironically we were allowed to spray everywhere oxytetracycline, and I haven't seen working in anywhere, honestly. So, so it is very hard to believe that what you are saying, spraying on the trunk, is going to do any different. But I like to see data, honestly. So far, all our experience indicates opposite of that. It's oxytetracycline is quickly breaking down by the UV, and it's you know,
Well, we're talking about trees this big that don't take very
small
long to take it up.
trees.
Yeah, maybe it diffuses.
But,
I don't know, maybe yes.
I mean, you
but-
do the same thing with your nati.
Yeah, in the greenhouse it works. Whatever you do.
As an alternative to IPCs, you know, I'd like to see y'all try that and just see. I mean, there's some UV inhibitor.
Yeah, I would think you can develop something like this too. Yeah, maybe some company will come up if they're interested in citrus. The problem with companies is citrus is not such a lucrative
It's true. Yeah,
crop, not a row crop or something, you know.
that's true. Unfortunately.
I would comment a couple of things. I was at a private trial that's been going on for 4.5 years or better. Many different rootstocks, many different varieties that she has presented as true. It's amazing. I've seen the most beautiful young tree. I don't think I've ever seen one as pretty in my life that was injected.
You were injecting already?
After IPCs, they did the IPCs, probably close to what you were saying, the 2-inch trunk. and then they injected them. Wow. I mean, and then they had trees up to 20 years old, you know, had 4 or 5 boxes on the tree, plenty of bricks, you know, just one of the trees, I was amazed. It was obviously, it had phytophthora around the base of the tree, you know, almost black hearted, and it was healthy as an ox. Other than that, big fruit, a lot of sugar. I think you're going to see some good results with this.
I hope, yeah, I just hope that everybody's doing the right thing. You know, you can do a lot of bad things probably with injection. Yeah, just yeah. Well, it's the best thing we have at this moment. It's kind of sad. It is what it is, right? So try to use it consciously. So do it as properly as possible.
Okay, guys, thank you very much. It's getting
Thanks
late.
for coming.
Thank you, Ute. Thank you,
Thank you,
Fernando.
Mongi, for organizing.
Thank you, Alison. Thank you all for participating. If you are in Zoom and you are interested about CEUs, send me an email.